Now that we know about the three types of carbohydrates, mono, di, and polysaccharide, we're going to spend a little bit of time here talking about the three most common types of polysaccharides. These really big sugar molecules. These sugar molecules made up of two or more monosaccharides. First one is starch. Second, glycogen. And the third is called cellulose. Starch is only found in plants. This is how a plant stores its extra sugar. All right, plants, just like you and I, use sugar for energy, and when they have excess of it, they store it as starch. And again, starch is a polysaccharide, so that means there are many monosaccharides, aka glucose molecules, singular sugars, bonded together. Glycogen is only found in animals. And this is how we store our excess glucose, because almost always there's excess glucose because we break it down so quickly and use it for energy to complete the functions necessary for life. Our liver actually bonds together many molecules of glucose to form glycogen. So our liver takes extra glucose in our body, binds them all together to make a polysaccharide, and we call that polysaccharide in humans glycogen. And lastly is cellulose. Cellulose is like that stringy stuff I showed you back on the carbohydrate functions slide in regard to what carbohydrates can be used for, quick energy and structural features. So cellulose was that white stringy stuff that was being used as a structural component of plant cell walls. It really gives strength and rigidity to that plant cell wall. And also, if you're taking notes on paper, or you've ever worked with wood in the past or present or are going to in the future, cellulose is a large component of paper products and wood because plant cells make up those objects, and oftentimes they're dead plant cells. In fact, all the time they're dead plant cells. So all that remains is the cell wall, comprised mostly of the polysaccharide cellulose. Now we're going to get into the new stuff, the macromolecule known as a lipid. Macromolecule known as a lipid. Some examples of lipids are fats, oils, and waxes. A more common name for a lipid is a fat, and you can see I have some fats, oils, and waxes displayed here on the side for you. Fats are generally not soluble in water. This means they're not going to dissolve in water. All right, water, the universal solvent, cannot dissolve lipids because lipids generally don't have a charge. And if a chemical doesn't have a charge, water can't bind to it, and therefore water can't dissolve it. And just like carbohydrates, lipids contain three different elements. All lipids are only made of these three elements, aka atoms: carbon, hydrogen, and oxygen. The same three that comprise carbohydrates. But you're probably asking yourself, well, Mr. Reese, what the heck is the difference between lipids and carbs? If they're made of the same atoms, aren't they just the same thing? Just like when it comes to protons, neutrons, and electrons making up atoms, it's the quantity and the distribution of these atoms within these macromolecules that determines whether it's a carbohydrate or a lipid. Back when we were talking about carbohydrates, I told you that these atoms, carbon, hydrogen, and oxygen, are always seen in a 1 to 2 to 1 ratio. But we don't see that same ratio in lipids. We do not see a 1 to 2 to 1 ratio in lipids. We only see that in carbs. All right. So the fact that we don't see this ratio in lipids gives these macromolecules different properties that carbohydrates don't have, so therefore they are different. The two building blocks, the monomers technically, of lipids are fatty acids and glycerol. Fatty acids and glycerol are the monomers of the polymer, the much bigger molecule we know as a lipid. And we call this giant molecule a macromolecule. All right, so a lipid has one molecule of glycerol and then 3 fatty acid tails attached to it. It looks a little bit like that. Okay, so here is your glycerol, this big guy right here, this long structure. Okay, and then each one of these is considered a fatty acid tail. All right, each one of these is a fatty acid tail. Now notice there's a difference between these two images. Here's your glycerol again. Okay, here's your fatty acid tails. But notice in this lipid, all the tails are straight. But in this lipid, this tail is kind of curved off to the side a little bit. All right, and that's because of this bond highlighted in green here. This is what's known as a double bond. We saw this back when we were talking about carbon compounds at the beginning of macromolecules. Carbon can form double bonds between itself and other molecules, and here, or other atoms. And here we see a carbon-carbon double bond. And this double bonds are very firm, very rigid, very hard to break. And because they're so rigid and strong, it actually causes a curve within the fatty acid tail. So there's our glycerol on both, and then here are our fatty acid tails. Okay? Now these are different types of lipids, but it's still a lipid, so it's only comprised of a glycerol and then 3 fatty acid tails.